Aloha everyone! Today we are going to be starting chapter 12, in which we're going to be learning about the brain itself. So back in chapter 11, we learned a little bit about the brain, right? When we talked about neurons, but we really didn't learn everything about the brain. So the brain is what makes our bodies function, and there is a lot to go over with the brain, so we are going to be doing that in chapter 12. So this is chapter 12, part A. We're going to be talking about the central nervous system. Today we're going to be focusing on brain development, and then we'll go into some of the anatomy and physiology of the brain as well. In terms of our learning outcomes for this lecture, first of all, we're going to describe the major functions of the nervous system, which of course is going to be linked to the brain, right? And then we're going to list the 5 developmental regions of the brain. And we're going to kind of look at how everything starts as a fetus and how that changes to the major areas of the adult brain that are going to arise from these 5 developmental regions. So if you haven't seen before what our brain starts as, it'll probably be, well, mind-blowing for you, because it's really cool how it starts as a neural tube and then comes to be our amazing brain that we have. So because we are talking about the brain, that means that we are going to be focusing on the central nervous system, or the CNS, right? And as a reminder from what I mentioned in the last lecture, the central nervous system consists of the brain and the spinal cord. You can see here that we have cephalization. And maybe if you've taken a zoology course, you've heard about cephalopods. So cephalopods are things like octopi, right? Squid, and they have a central nervous system. What that means by being cephalized is that it has a brain and then neurons that reach out to the muscles. So that is evolutionarily developed as a development of the rostral. Rostral just means the same thing as the anterior portion of the central nervous system. And this resulted in a major increase in the number of neurons. That's why we have so many neurons in our body. As a reminder, neurons are going to be those messengers that deliver information from the brain to the other parts of the body. And as we progress through evolutionary time, organisms started to do more things, right? We were much more, organisms started to become more specialized. If you think of a sponge, then think of an octopus, then think of a whale, then think of us, right? We're adding things through evolutionary time, right? So started to do things like walking on all fours, going from walking on all fours to walking on 2 legs, right? And then humans can talk, right? We started to listen a lot more. We started to gain a lot more information. So we developed a lot more neurons to be able to do all of those things. Thus, we needed to create more space. So the highest level of the central nervous system, as we know on Earth, is actually reached in the human brain. So if you didn't feel special before, hopefully you feel special now, because our brains are pretty amazing. So as I mentioned in this lecture, we're going to look a little bit at brain development. So how did our brains become so amazing? How did this brain development happen? Well, the brain already starts to develop right after conception. It's actually the first thing that starts to develop before everything else. So here you see what our brain begins as. And as I told you, it looks pretty different from what we're used to seeing of an adult brain, right? So this green and yellow tube-looking thing here is the brain and the spinal cord. This is known as our neural tube. And there's the anterior, and I told you rostral is another term for that. And then there's the posterior, or this caudal area. And this develops in the fetus roughly 4 to 6 weeks after the woman's last menstrual period. So this is just a really, really thin tube. And many times, a woman may not even know that she's pregnant when this is already developing, right? So 4 to 6 weeks after that last menstrual period. The brain and the spinal cord, right here, they begin as this neural tube. So what's different between this neural tube and the adult brain? How are we going to go from this to what we see on the next slide? So just to kind of reorient you again, we're starting out with this neural tube, right? And I asked you, how is this different from the adult brain? Well, just a few differences, right? It's very different. The neural tube is very different from the adult brain. It grows so much. You can see in the brain there are a lot of different wrinkles. We'll talk about why those are there. There are a lot of different crevices, a lot more tissue. So that neural tube eventually becomes the adult brain. So we're going to be talking about this process of brain development in these next few slides of how we go from this neural tube to the adult brain. So how do we get there? We're going to look at the embryonic development of the human brain. We're going to start from that neural tube that I showed you before and go to these primary brain vesicles. So how do we go from neural tube to the primary brain vesicles? So first, again, just to remind you, that neural tube right here, it's going to develop about 4 to 6 weeks after the woman's last menstrual period. But then the neural tube is going to expand anteriorly. So this anterior portion, you can see how it's extending and expanding there. And it's going to form these different constrictions. And what I mean by constrictions is, notice the width is not the same all the way down, right? You can see it goes in, back out, in, out. That's what I mean when I say constrictions. And by forming those constrictions, we're kind of forming different regions. So specifically, we go from this neural tube to having 3 different constrictions. And it gets sectioned off into 3 primary vesicles. We have the prosencephalon right here at the top, most anterior region, that's the forebrain. Then we have the mesencephalon, and anytime you hear mes, it generally means middle. So this is going to be our midbrain in the middle right here. And then we have the rhombencephalon. This is going to be the hindbrain. So we have these 3 different primary brain vesicles. So that happens a little bit after that neural tube develops. As you probably guessed, we don't just end with these primary brain vesicles though, right? That still doesn't look like the adult brain. So then later on, we start to differentiate these different primary brain vesicles into secondary brain vesicles. And there are 5 different types of secondary brain vesicles. So this forebrain region here, it's going to develop into 2 different regions. The telencephalon right up here. It kind of looks like Mickey Mouse ears, if that helps you to remember it. And then you have underneath that the diencephalon. So the prosencephalon becomes the telencephalon and the diencephalon. And the telencephalon is going to become really a lot of that main forebrain area that we're going to be talking about. The diencephalon is going to develop into things like the hypothalamus, which we'll talk about in another lecture. Then this mesencephalon, notice how it gets a little bit bigger, but we don't break it into any other vesicles, right? The mesencephalon stays the mesencephalon. And that's the only part of the brain that does not change from the primary brain vesicles to the secondary brain vesicles. Then lastly, we have this rhombencephalon, or the hindbrain, and it's going to differentiate into the metencephalon and the myelencephalon. So we can see these different vesicles here. They're arising due to enlargement, right? It's larger than it was as the primary brain's vesicles, right? So it's getting larger, and then we're also having more of these constrictions, right, to make those different vesicles. So that helps us to denote the differences between the different areas, shows us that now we have these secondary brain vesicles. And what's really cool is that those secondary brain vesicles right here that we just talked about, they're going to develop into these adult brain structures and adult neural canal regions as well. So the Mickey Mouse ears, that telencephalon right here, that's going to give rise to the two cerebral hemispheres that together make up our cerebrum, which is basically the largest part of your brain. Then this diencephalon is going to become the thalamus, hypothalamus, epithalamus, and the retina. So that's all the posterior regions of your brain that are controlling a lot of your emotions and more complex parts of movement. Then we have this mesencephalon, or that midbrain. That's going to become your brainstem, right? We also have portions of the metencephalon and the myelencephalon that you can see also are going to form part of that brainstem as well. So the mesencephalon is going to remain that midbrain region. The metencephalon is going to become the pons and the cerebellum. And then the myelencephalon is going to become that medulla oblongata. The central cavity of this neural tube, so the central portion here, central as you go throughout the development here, that becomes the ventricles. And finally, the most posterior region of this neural tube, so down here, down here, down here, right here you can see it in yellow. You guessed it, it becomes your spinal cord. And it's going to travel all the way down from our brain, down our spine, and the next picture is going to show how exactly that development is going to work. So I really like this series of pictures here because hopefully it gives you a better idea of how exactly we go from that neural tube to the primary brain vesicles to the secondary brain vesicles to our adult brain. So this is what's happening when a child is growing. And you can see how they develop the primary vesicles, right? The prosencephalon, mesencephalon, rhombencephalon, you have that spinal cord, right? So that's up here. You can see kind of how you have those primary vesicles curved there. Then they're going to develop the secondary vesicles. So these are those secondary vesicles, right? Your telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. So you can see each of those here in this picture. And those secondary vesicles start to grow, and they develop into the adult brain. So why does it matter that our early brain is going to make a bunch of folds and crevices? Like you can see how this isn't straight across, right? You can see how it's kind of folding back on itself. So it's because it needs to fit all of this, our whole brain, inside of our head. So in order for the brain to develop, it needs more space, right? The primary vesicles are going to start to kind of curve in on themselves and then starts to grow up over. And you can kind of see that in these images, right? You can see how it's kind of growing over on top of itself, kind of folding, bending, just to fit in that space. The telencephalon actually ends up covering everything and overlapping on top. So this telencephalon here, you can see how it's starting to overlap these regions, and eventually it completely does an overlap here. So the telencephalon ends up covering everything and overlapping, and that's because it starts to fold. And the more it folds, the more surface area that allows for more neurons to grow. So the brain grows a lot faster than our surrounding membranous skull. And in order for the skull to keep up with our brain has to fold. So usually the forebrain, as you can see in the dark purple here, it starts at the front end of the head, the anterior portion, but then over time it's going to need more space and it grows almost everything. That's why you almost see all purple here, right? And this ends up being the final product that you see here, right? The forebrain moves all the way towards the brainstem and actually develops in the superior part of the head, and the cerebral hemispheres are all going to double back and envelop the diencephalon and the midbrain, while creasing and folding to increase that surface area. So kind of crazy still, even once you see these different pictures, right? How we can go from that little neural tube to the primary brain vesicles, the secondary brain vesicles, and then get to this part where we actually have our brain here. So a lot of folding is what allows that to happen. So I just want you to keep in mind that the increased surface area or structure of that brain is what gives humans immense neurological function. It was our ability to grow our brain and fold our brain in this way during our embryonic development that gives us a lot more evolutionary advantage. It gives our brain an incredibly unique structure, and it allows for the development of millions of neurons to be able to make the connections that we need to have such a highly developed brain. So really cool. I know I'm kind of talking up our brains a lot, but hopefully you already know how amazing our brains are. So now that we've talked about that development from the initial neural tube, this is our lovely adult brain. So these cerebral hemispheres, you can only see one of them obviously because we're not looking at it in 3D, but here's that cerebral hemisphere, there's one on the other side. So the cerebral hemispheres grew over everything. It grows over that diencephalon, and it breaks up into 4 main parts, which we know as the cerebral hemisphere. That's going to be the main part of the brain, right? The diencephalon, right here in purple, is kind of located in the center. Then we have the brainstem, and the brainstem is going to include our midbrain, your pons, and your medulla oblongata. So everything right here in green is your brainstem. And then we have the cerebellum right here in the back or posterior portion. So those are kind of the 4 main parts of the adult brain. So now we're going to be talking a little more about the anatomy as well as the physiology of the brain. So when we look at the brain regions and organization, adult brains are going to have 4 main regions that consist of either white or gray matter. And they surround the central ventricles. So we have the cerebral hemispheres, the diencephalon, that brainstem, and the cerebellum. So these are those 4 different regions that I showed you on the picture in the previous slide. And if you remember, we talked about white and gray matter last chapter, and they make up all of these parts of the brain. And like I showed you in the previous picture with the adult brain, there's those cerebral hemispheres, your diencephalon, that brainstem, which again has the midbrain, the pons, and the medulla, and then the cerebellum. So we're going to be talking about all of these today, and we're going to talk a little more first about that white and gray matter just as a review from our previous lecture. So when we look at the brain regions and organization and that differentiation between white and gray matter, just as a refresher, gray matter is going to be short, non-myelinated neurons and cell bodies, whereas that white matter is going to be myelinated, and there can be some non-myelinated axons as well. So gray matter is going to be all the non-myelinated neurons, and this means that there are no myelin sheaths on the neurons. And they're usually going to be shorter, right? Because they're not going to be able to transmit that signal as far, right? Whereas with the white matter, the axons, those can travel really long distances, right? Because they're able to do that conduction where they're jumping from node to node with that myelin there. And their axons are going to be myelinated most of the time. Unless that white matter is just going a really short distance, then it's usually not going to be myelinated. So the basic pattern that we see in the central nervous system is that there's going to be a central cavity, and that cavity is going to be surrounded by the gray matter, with the white matter external to the gray matter. And we're going to see some exceptions to this rule and how it kind of differs in the central nervous system, but this is the basic pattern that we see in the central nervous system. So as I mentioned, we have that basic pattern, but we have some exceptions to that, right? So here are some of the features of the patterns and the change in the pattern that we see with the gray and white matter in the central nervous system. So the spinal cord is going to exhibit that basic pattern of the gray matter inside and the white matter outside. However, that pattern is going to change with ascent into the brainstem. So when you go into the brainstem, it's going to change just a little bit. So in the brainstem, we're going to have additional gray matter scattered within the white matter. So the cerebral hemispheres, cerebrum and cerebellum, the back part, we're also going to see things change a little bit with those as well. But in terms of the spinal cord, what we see is that that gray matter is usually in the inner part. And I'm going to back this up and kind of follow my bullets. I was kind of trying to tell you everything at once. So I'm going to back this up and go nicely through the bullets. So spinal cord, basic pattern, right? Where we have the gray matter inside, white matter outside. Then we have the brainstem. What we see with the brainstem is that we're going to have some additional gray matter nuclei. It's going to be scattered throughout the white matter, and I'll show you that on the next picture. Then in the cerebral hemispheres, so that's going to be the cerebrum and the cerebellum, they're going to have an outer layer of gray matter called the cortex. And the cerebrum and the cerebellum, just like the brainstem up here, are also going to have different areas of gray matter scattered around the white matter. And then in the spinal cord, the gray matter is usually in the inner part and the white matter is in the outer part. So it kind of flips, right? In terms of in the brain itself, in that cerebrum, the gray matter is outside, the white matter is inside. So we see kind of a flipping, and I think all of this is going to make a lot more sense when we look at the pictures on the next slide. Okay, so I apologize, that was maybe a little bit confusing. I think it'll be a lot better when we look at these pictures here. This is how it looks. So here is our spinal cord region, right? And what you're pretty much seeing is just some cross sections through different areas to look at the white and gray matter there. So here is that spinal cord region, that cross section from it. And what you see is that gray matter. So gray matter right here, it kind of looks like this butterfly. So that's going to be on the inside, right? Then the white matter surrounds it. We see that in our spinal cord. Then when we move up into the brainstem region, it's still kind of the same. So if we take this cross section here from right here, this is the brainstem area. So we can see that we still have this inner gray matter. We have a lot of this outer white matter. But the thing that changes a little bit is that we kind of see scattered throughout this white matter are some areas of gray matter, right? So it's just kind of scattered throughout that white matter, but we still see that basic pattern of gray matter inside, white matter external to that. Then when we get up to the cerebrum and the cerebellum, so this is a cross section right here through that cerebrum portion, it looks a little bit different, right? So we have the gray matter on the outside. And this outer portion of gray matter, this is known as the cortex of gray matter. So everything that you see out here, it kind of looks like an outline around, that's the cortex of gray matter. Obviously, we don't see that in these other ones, right? That's something different. And the cortex of the gray matter is going to protect the brain. You can see that there's also white matter right inside of that cortex. And then you have more of this gray matter in here, more internal, right? So usually the gray matter is inside, white matter is going to be outside. In the brain, it's just that there's that cortex of gray matter, this cortex right here, that's layering the outside of the brain and makes it kind of look like it's flip-flopped, right? So the neural information is usually going to originate in our gray matter, and then it moves to the white matter. So the white matter is where that neural information travels. So everything is kind of originating from the center. So gray matter is where it originates, goes to the white matter to travel out. So kind of an analogy to give you, gray matter is kind of like your driveway or your carport. That's where your car starts, right? And then the white matter is going to be the road or the highway. That's how it actually travels throughout. So hopefully this makes a little bit more sense. You can see this is the basic pattern of the central nervous system in terms of white and gray matter, where we have inner gray matter, outer white matter. Brainstem, we still have that pattern, but we have some scattered areas of gray matter. Then in the cerebrum, we're going to have islands of gray matter within that white matter, and then this outer gray matter cortex. Okay, so moving on, in the brain, there are different regions. And as you can see, they're colored in different colors here. And they are going to contribute to different things. So some terms that I want to introduce first, before we start talking more about the anatomy of the brain, is the gyri. So you can see if it's just singular, it's called a gyrus. So a gyrus is a ridge of the brain. We also have a sulcus. A sulcus is going to be a shallow process or a shallow groove on the brain. So if you look even deeper, you may see some fissures. So here's an example of a fissure. And the fissures are just really deep grooves in the brain. And the reason why there's all these grooves is, again, to pack more of that gray matter into the skull. So that's why our cerebral cortex is wrinkled, right? We're trying to fit all that in a smaller space. So again, the gray matter is the area where we produce that information, like your driveway or your carport. And then from there, if we produce fissures, then we can transmit that information to our white matter a lot better. So in this bottom left photo here, you're seeing the cerebral cortex right here. It's that gray matter, right? It overlies this cerebral white matter, right? It's about 2 to 4 millimeters thick, containing 20 billion cells. Crazy amount. It has about half of all the neurons in the human brain. And beneath this cortex is going to lie that cerebral white matter, right? So gray is that cortex, then you have that cerebral white matter. And the tracks that connect the parts of the brain with itself and other parts of the nervous system are going to lie in this white matter because, again, that white matter is like the highway or the road, right, that allows things to transmit out from that driveway or carport. Additionally, we have 4 different ventricles in our brain. So we have 4 cavities that are filled by cerebral spinal fluid. There are paired lateral ventricles. So you can see that here, your lateral ventricle. And this is showing you the anterior view of the brain. This is showing you that left lateral view of the brain. But we have paired lateral ventricles that are going to connect to the 3rd ventricle via the interventricular foramen by the septum pellucidum. So if you look here, your lateral ventricle connects to this 3rd ventricle right here via this interventricular foramen, right? And we have the septum pellucidum right here as well. So it's going to connect right there to get to this 3rd ventricle that we see right here. So we have, told you there are 4 total, right? We have 2 lateral ventricles, we have the 3rd ventricle, and then we have the 4th ventricle. So the 3rd ventricle in the diencephalon region is going to connect to the 4th ventricle right here, and it connects via the cerebral aqueduct right here. So you can see that connection between the 3rd and the 4th ventricle. So the 4th ventricle in the hindbrain is going to connect to the central canal of the spinal cord and the subarachnoid space via the apertures. And then it goes back via the dural vein sinuses via that arachnoid granulations. So you can see that here. This 4th ventricle is going to go down here into that spinal cord region. So each of these lateral ventricles, just as a little bit of review, so you have two lateral ventricles, right? They're going to be connected to that third ventricle via this interventricular foramen. So I want you to remember that. And then you have your third ventricle connecting to the fourth ventricle via this cerebral aqueduct. So those are your four different ventricles that we have in the brain: the lateral ventricles, the third ventricle, and the fourth ventricle. So next we're going to talk a little bit about the hemispheres. We're going to look at the different cerebral hemispheres. Cerebral hemispheres are going to form the superior part of the brain, and they account for about 83% of our brain mass, which is the majority, right? So the surface markings that we can see, I mentioned some of these a little bit earlier, but we can see gyri. You don't really have any of those pointed out in here, but just as a reminder, the gyri, those are going to be ridges, right? Fissures are the really deep grooves. You can have longitudinal fissures. So your longitudinal fissure actually is here. So you can see what that longitudinal fissure does is it's going to separate the two hemispheres of the brain, right? So you have two of those cerebral hemispheres. This longitudinal fissure separates them. You also have the transverse cerebral fissure. You can see that over here in this left lateral view. And you can probably guess what that fissure is separating, right? It's separating the cerebrum from the cerebellum portion. So again, those fissures are just deep grooves. Two of them that I need you to know are this longitudinal fissure and this transverse cerebral fissure. Okay, so we have those two cerebral hemispheres, but it gets a little bit more complicated than that because those hemispheres can be divided into 5 different lobes. There's a frontal lobe, a parietal lobe, a temporal lobe, an occipital lobe, and the insula lobe. And a lobe is just a rounded projection. So next I'm going to show you where each of these different lobes are on an actual picture, and we're going to talk about why they're important. So here you can see the lobes, the sulci or the solstices, the fissures of the cerebral hemisphere. So several sulci are going to divide each hemisphere into 5 different lobes, like I mentioned on the previous slide. So first is going to be this frontal lobe, which is just like what it sounds, right? It's in the front or that anterior portion. So it's here in purple. The frontal lobe is, of course, in the front. Then right behind it is going to be your parietal lobe in red. Then the occipital lobe is right posterior to that right here in the light blue. Then underneath the frontal and the parietal is going to be this temporal lobe, which you see in orange here. So that's your temporal lobe. And these 4 lobes, if you remember back to when we learned some of the different bones of the skull, the 4 lobes are actually named after the overlying skull bones, if that kind of helps you to remember them. But I only mentioned 4 lobes, right? The reason for that is that below everything is going to be that 5th lobe, which is the insula, which we cannot see from the outside here. And as I mentioned, you have these sulci, that's what all these are, that are actually dividing the cerebral hemisphere into these different lobes. And then down here, this is just kind of to illustrate again how these different portions of the brain are named after the overlying bones. So frontal bone, frontal lobe, parietal bone, parietal lobe, occipital bone, occipital lobe, then your temporal bone, temporal lobe. So hopefully that helps you to remember those names. I mentioned that in the previous slide we could not see the insula, but here we actually can see the insula. So in this figure, you can see how there are these two tongs kind of pulling apart the brain. And if you pull apart the brain, then on the inside, right here, is where the insula is located. So the insular lobe is this inner area that is buried underneath the portions of the temporal, parietal, and frontal lobes. Next, we're going to look a little bit at the sulci and fissures of the cerebral hemispheres. And as a reminder, sulci or sulcus, that means your valley. So those are going to be the groups. And gyri or gyrus means mountains. So those are going to be the ridges on the brain. And then the fissures are kind of like when you have an earthquake and it creates really large cracks in the ground. So that's just a very deep type of sulcus. So here's a picture to show all the different lobes, of course, except for that insula, which is going to be inside, right? I showed you that on the previous image. So some of these that I would like to point out to you, some of the major sulcus that divide the lobes. We have the central sulcus right here. And as you can tell, it is dividing the precentral gyrus of this frontal lobe in red from the postcentral gyrus of the parietal lobe here in blue. So dividing that frontal and the parietal lobe is that central sulcus. And I meant to put boxes around these, so if you're able to, go ahead and make sure you're circling these different ones that you should know. Then we have the parietal occipital sulcus. This is going to, you can see it right here. This separates the parietal lobe from your occipital lobe. Then we have the lateral sulcus right here. That is going to outline your temporal lobe, which is in this dark blue, purplish color. And then the cerebral, as a reminder, you have that cerebral cortex of gray matter superficially. That's what this is kind of meant to show you. That cortex of that gray matter is superficial. And then underneath that, you have that white matter, which we already talked about earlier in this lecture. In terms of our cerebral cortex, it is really important for all of our functions. It's in charge of things like our awareness, our sensory perception, voluntary motor initiation, communication, memory storage, and understanding. So the cerebral cortex is the executive suite of the nervous system where our conscious mind is found. It makes up about 83% of our brain, and I just wanted to include this picture here because this was in your book, and if you didn't know, scientists are able to use MRIs, so magnetic resonance imaging, to scan where the blood flow is in your brain when you're doing different things. So they can scan someone's brain, that's what this is from, and you can see what their brain looks like when they're doing different functions. So when they were focused on seeing things and visually being stimulated, they noticed that that blood flow is more in the frontal lobe. And then they noticed that when you're trying to listen in your hearing, it's more in that central part of the brain right here. So where it's really red and orange, that's showing you where that blood flow is. Speaking is going to be more towards the posterior end of the brain, and thinking, you can see, is kind of distributed all throughout. And I just thought it was a really interesting picture showing the blood flow in the brain. Because again, when we talked about neurons, neurons are supplied with a lot of blood flow, and also the neuron transmission, that's associated with a lot of heat, right? A lot of heat is generated in that. So that's how we're actually able to understand which part of the brain contributes to which function. That's how we know the physiology of our brain, which is pretty cool. So we're going to talk a little bit more about the cerebral cortex and look at 4 general considerations of the cerebral cortex. So the cerebral cortex contains 3 main types of functional areas. There's a motor area that controls our voluntary movement, such as moving our arms, moving our legs. There's sensory areas, which control our conscious awareness of sensation, like touch, sight, sound, etc. And then there are association areas, which are pretty much going to combine all of that. So it's a very complex thing of taking in that information and then putting it out to the body. But I'm going to be talking about each one of them, the different motor, sensory, and association areas in this lecture. Also, for the cerebral cortex, each hemisphere is going to be located with a contralateral side of the body. What that means is that there are two hemispheres of the brain, which we already talked about, right? And if I want to move the right side of my body, that means that the left side of the brain is what's going to function. On the other hand, if I'm going to move the left side of my body, then the right side of my brain is going to function. Also, we have lateralization of the cortical function, and that can occur only in one hemisphere. So what that means is only certain parts of the brain are going to work for certain things. And if one part of the brain is disrupted, then that's going to contribute to a specific function. We're going to see something go wrong with the function, right? Lastly, what I want to mention here in terms of these four general considerations of the cerebral cortex is that conscious behavior involves the entire cortex in one way or another, because the brain kind of all works together to transmit information from one part to the next. So I'm going to actually show you this picture in a later lecture too, but I just wanted to show you it here as well. You might have seen this before, but again, there are different parts of the brain that function for different things. And someone has illustrated this really nicely to show a generalization of what the certain areas of the brain are linked to. So there's certain parts of our brain that are linked to our lips and our jaws and our tongues, and this is totally broken down based on just even the slightest area. So you'll notice if it is more anterior, then it's going to be more the motor information. So this is the motor map. This is more that anterior portion of the brain. Whereas if it's more posterior, then it's more of this sensory information. So we're going to talk more about this. We'll label all this later, go more in depth with this figure. But I just wanted to show you this as kind of a preview of how really cool our brain is. Okay, so I already kind of alluded to the fact that we're going to be walking through these different lobes of the brain and talking about their anatomy and their physiology. So first, we're going to be covering the frontal lobe. This is the front part of our brain, and it's mostly about the motor areas or the movement in our body. And motor areas are going to act for controlling our voluntary movement. Voluntary, again, meaning you have to decide to do it, right? I have to decide to lift my arm up. My arm just doesn't constantly lift up and down by itself, right? That's what voluntary means. So here is our primary motor cortex right here, this portion right here. And we also have our premotor cortex right next to it right here. And we have our Broca's area. It's the other thing I wanted to mention. You can see Broca's area right in here. So Broca's area is kind of this dotted line portion around the outside of it. And we also have our frontal eye field region right here. So all of these different areas, obviously, different form, so they have a different function, right? So they're going to function in different things. The primary motor cortex, right here, this is actually located in that precentral gyrus, right? Because it's right before that central sulcus that actually separates it from this lobe right here. So this is that primary motor cortex, and it is mainly involved in primary movement, like our arms and our legs. So it's pretty much the first things that come to mind when you think of your primary movements. Then we have the premotor cortex, which is anterior to that precentral gyrus. So it is more so in complex things like playing an instrument or typing. So it takes a little bit more of that fine motion, fine motor movement. Then, of course, the frontal eye field that I pointed out, it is within and anterior to that premotor cortex and also superior to Broca's area. It's going to be functioning in what we take in. And then the Broca's area, right down here again, where those dotted lines are, it is going to be important in special motor speech. So this part of the brain is usually going to, your Broca's area, it's usually going to activate before we talk. So as we're thinking of what we're going to say before we actually say it, it's the Broca's area that's going to light up in those MRIs. So that's the functions of these 4 different parts of the main motor areas of the frontal lobe. So again, primary motor cortex right here, your premotor cortex, your Broca's area, and your frontal eye field. Again, I did not put boxes around these, so make sure that you know those 4. Obviously, there are a lot of different things in this image. I want you to focus on these 4 right here. Those are your primary motor areas in this frontal lobe. So this right here is that premotor cortex again, and it's more for those fine motions. And then there's the primary motor cortex. So if I want to type, then I'm going to send that information to my primary motor cortex, which contains all of my main information to my muscles, right? Then it's going to send that information down my spinal cord, right, to go through my nerves all the way down to my hands to type. So that's how the information travels from one area to another. The premotor cortex is usually going to select basic motor movements and put them into more complex tasks. So it uses the functions of the primary motor cortex to control those voluntary actions. Because in order to move my fingers a certain way, like if I want to move my finger up, I need to take that information through my arms and down to my fingers. So that's why different parts of the brain are going to communicate to each other in that way. So the premotor cortex is going to be helpful in planning the movements. It's kind of like a staging area for the skilled motor activities. It contains a lot of our learned repetitions or patterned motor skills, and it controls a lot of the voluntary actions that are going to require sensory feedback. And then those portions of the body are going to be made up of what we term pyramidal cells. And these are just large neurons that allow conscious control of precise, skilled, and skeletal muscle movements. So these regions are made up of pyramidal cells. You can see that it's talking about the pyramidal tract. So it's made up of pyramidal cells that go down this pyramidal tract right down here. So this again is just that motor portion of the brain. And of course, we have one of these lovely homeostatic imbalance slides. I really like these because they give you kind of a clinical perspective on, okay, we're learning all this information, what can go wrong with that? So what would happen if we were to damage our primary motor cortex? This actually usually happens when someone has a stroke. And when you have a stroke, you have a blood vessel burst, and then it paralyzes your muscles that are controlled by those areas. Because now we can't send that information from the motor area to the parts of our muscles. We can't control them. So that's why when someone undergoes a stroke, usually that result from that is going to be paralysis. So again, damage to the areas of that primary motor cortex as seen in a stroke is going to paralyze the muscles controlled by those areas. Paralysis is generally going to occur on the opposite side of the body from the damage, which makes sense because we talked about how if you want to do something with the right side of your body, it's going to be the left side of your brain that allows that to happen, right? So if you have damage on the left side of your brain, that's going to affect the right side of your body. And you may have heard these two different, there are more than two, but these two different types of strokes. So you can have an ischemic stroke. That's where a clot is going to block the blood flow to an area of the brain. So you can see here, it's not able to get up into the brain, right? It's blocked. Whereas with a I always mess up this word, a hemorrhagic stroke, it kind of sounds like a hemorrhage. This is bleeding occurs inside or around the brain tissue. And it's pretty much causing a hemorrhage of that, and that's why it's called a hemorrhagic stroke. So difference there from your ischemic stroke, where you can just see that clot is blocking blood flow into the brain. I say just, it's a big deal, right? We need that blood flow in our brain for our brains to function. So looking a little more at Broca's area, and I already talked a little bit about Broca's area. You can see, again in this image, how it's kind of outlined by these dashes. And Broca's area is going to be helpful in speech. So it's controlling the muscles of speech production. But what's unique about it is that it's usually only present in one hemisphere, which is the left hemisphere. So if the left part of the brain, that left hemisphere is damaged, then of course we can't talk, and our speech is going to be impaired. So that's a big thing that we also see happening with strokes too, right? A lot of times if that happens in the left hemisphere, we'll see speech is severely impacted as well. But it's not just Broca's area that is important for our speech. So there are other parts of the brain that actually interact with Broca's area. One of those that's really important is Wernicke's area. And it's going to comprehend and formulate speech. Whereas Broca's area, from that frontal lobe region, is going to take and receive the information, takes and receives it from Wernicke's area, and then sends those impulses to the premotor area to go then to the motor area, and then it goes to our mouth to actually regulate our tongue and jaw and our whole mouth movement. So if any of those areas along that whole path are damaged, then we can't speak. So again, it's going to be Wernicke's area that is taking in that information, and it formulates the speech. And then Broca's area takes that information and sends the impulses and the neurons down to the premotor area. So essentially, Broca's area is going to be associated with language production, while Wernicke's area is associated with language comprehension. And you can see in this bottom image, so you have Broca's area right here, you have Wernicke's area over here. They're a good distance apart, right? Well, they're actually connected by arcuate basilis right here, which is, we've talked about these before, it's essentially a bundle of nerve fibers that actually connects the two together so that they're able to work together. And then up here, you're also seeing your Broca's area here in purple. Wernicke's is in this bright green. This is your primary auditory area. So you can see how it kind of starts here in this primary auditory area. Then Wernicke's area, again, trying to comprehend that language. Then we're going to Broca's area. Broca's area is going to be trying to work on that language production, sends that up to your motor area, to your premotor cortex, right? So that we can actually speak. So we talked about motor areas, right? Now we're going to go on to talking about association areas. So what exactly is an association area? This is going to be parts of the cerebral cortex that receive input from multiple different areas, and they kind of take all that information and output it. So association area just refers to anything that's not the primary motor or the sensory areas. Association areas are going to interpret sensory information and then form the connections between that sensory and the motor. So sensory motor association is kind of that link. It's the area of the cerebral cortex that links and coordinates the two together and helps in cognitive processing. So here you can see that prefrontal lobe right here. That's going to be your association area. We already talked about the motor areas right up here, right? We already covered those. Now we're going to be talking about here, these association areas. This is also known as the prefrontal cortex, and it is the most complicated cortical region. This area is going to be involved with things like intellect, cognition, recall, and personality. So it contains the working memory needed for abstract ideas to develop judgment, reasoning, persistence, and planning. So the development of this prefrontal cortex depends on the feedback from our social environment. You've probably heard of really sad cases where people will kidnap kids when they're really young, or they'll kidnap babies and then they just lock them up in a room, right, for long periods of time. And when that happens, those children, they're not able to develop their prefrontal cortex region because they don't have any outside information to take in new things and to develop their personality and their judgment and reasoning. Or even if a person has tumors in this prefrontal area or another type of lesion there, then it causes mental and personality disorders. You can get loss of judgment, loss of attentiveness, and loss of inhibition. There are even some people that maybe like to, they'll skydive without even thinking about the risk of it, or they'll do anything they can for their next thrill, right? And they don't even think about the fact of the matter, oh, I could die doing this. They don't care about dying. They have no really sense of danger at all. They're usually actually lacking some part of this prefrontal cortex because they don't have anything to inhibit them from improper judgment. So an infected individual like that would be oblivious also to a lot of different social restraints. They might become careless about their personal appearance and then again just take a lot of different risks. So that association cortex is going to be the cerebral cortex outside the primary areas that are essential for mental functions that are more complex than detecting basic dimensions of sensory stimulation. Okay, so now we're going to move on to looking at the sensory areas. These are going to allow for the conscious awareness of our sensations. And the sensory area, as you can see, is more in the posterior portion of the brain. The sensory areas are our senses, right? So it's conscious awareness of sensations like the five senses, your sight, sound, touch, taste, and smell. And these occur in the parietal insular region, which you can't really see, right? Because we can't see that insular region, right? Because it's underneath. Remember that picture with the tongs kind of pulling apart? The insula was underneath there. So we can't see that here. We also have our senses occur in part of the temporal region. So you can see that here. And also our occipital portion of our brain as well is going to be used for those sensations. So again, everything in kind of this posterior area, we're going to see more of that sensory processing. And I'll talk to you about all of these in detail, but just as a little bit of a preview, we have the primary sensory cortex. It's called the somatosensory cortex right here in this dark blue. It's in that postcentral gyrus region, right? It's going to be receiving the information from the skin to contribute to spatial discrimination. Somatosensory is understanding things like size, texture relationships. Visual areas are going to be interpreting images. So you can see here, that's your vision region. They're going to be interpreting images, color, form, and movement. Auditory, this hearing portion, right in here, that's going to be involved in pitch, loudness, and location. Vestibular is more for our conscious awareness of our balance. So you can see this vestibular cortex right here that's going to be in your insula. It's kind of your equilibrium. Olfactory is going to be for smell and odors, and it's not shown here. And gustatory is for your taste. You can see this gustatory cortex is also in the insula, so underneath here you can't see it. So we also have our visceral sensory, and if you think of visceral again, think of visceral organs, right? So like having an upset stomach, having a full bladder, or feeling like your lungs are going to burst, that's all going to be part of that visceral sensation. So you can see a lot of these different sensory areas here that we're going to talk more about in the next few slides. And here you can actually see a cut right down the middle of our brain, and you can see where that insula portion is, right? And this is what we cut through here. Anybody remember what separates those two hemispheres? It's going to be that longitudinal fissure. It breaks up the two cerebral hemispheres. So we have this central sulcus that I pointed out earlier. Basically, because it's breaking apart the frontal from your parietal here, it's breaking apart the motor from the sensory portions of the brain, right? And this just really shows you how small that space is. And those are two very, very different portions of our brain, right? So things like what we smell or what we hear, those are usually going to be discussed first with the frontal lobe. But then we can see things like our emotional reactions and our memories, they're going to be tied to our senses. So think of this as in maybe you associate a certain smell with your grandma's house, or you have a certain taste that you associate to a specific holiday memory. And senses are really cool. We're actually going to talk about them more in a later lecture. It's a chapter that we'll cover towards the end of this semester. So as I mentioned, next we're going to move into each of those sensory areas. And again, they're involved in sensation. They occur in the parietal, insular, temporal, and occipital lobes. And we get receptors, whether it's our light receptors from our eyes, our eardrums, or our skin receptors. Those receptors are going to go to the sensory cortex, which is the back area of the head. And then they go to the association cortex to distribute that information down to our brainstem and then to our nervous system. So as I mentioned, we're going to go through each of the different sensory areas, and we're going to start by talking about our auditory areas. Those are going to be the ones that pick up sound. So each sensory system has its own association areas on the cerebral cortex. And recall that the sensory systems, our vision, hearing, etc., they each have their own primary area on the cortex, which gets the most direct connections from its sense. Thus, all of these different colors here, right? So auditory areas, we have the primary auditory cortex. This is the superior margin of the temporal lobes. It's going to interpret information from the inner ear as pitch, loudness, and location. So that's going to be that primary auditory cortex, which you see kind of right here in this dark blue. Then we have the auditory association area. This is located posterior to the primary auditory cortex in this teal color. So right here, and it's going to store memories of sounds and permits perception of sound stimulus. So information from the auditory receptors, so you start with those receptors, they are going to go to the primary auditory cortex in this dark blue, right? It's this on that superior margin of the temporal lobe. That's where we hear the sound. And then that sound goes to the association cortex right here in this teal color, where we're actually recognizing what that sound means. So you see this picture up here of this mom talking to the baby. With babies, they have the neurons in the auditory association area are going to respond much better to complex sound patterns like bird calls and speech sounds rather than to simple pure tones. So that's why a lot of times they say that the more you talk to a baby, the better, because the more you talk to them, they're going to understand speech much better. The next sensory area I want to talk about is our visual senses. So we have information from the photoreceptors in our eye. They are going to transmit an image that goes to the primary visual cortex, which is this small red area in this occipital lobe right here. So that's that primary visual cortex. The information that the visual association area, so it's then going to go to this association cortex. The association area is in this kind of maroon color. It takes up much of that occipital lobe. So the information that it gets from the primary area is about simple contours, boundaries, edges, light and dark, and sensory qualities like color. So the visual association area on the lower part of that temporal lobe plays a primary role in your ability to do things like recognize faces, cars, trees, etc. Then we have nerve cells in a part of that visual association area that are going to respond to visual stimuli that have some sort of a complex pattern or a structure. So they usually only respond when our eye is looking at complex patterns, such as images of objects, abstract forms, hands, faces, or even specific faces to help us recognize somebody. So when we see a face, our visual association neurons are going to fire. So again, that's in this purple region, right, or maroon. And we recognize that face for something special and unique. And then when we see it again and again, we make those connections by repeatedly seeing the same complex structures. So again, for these visual areas, we have the primary visual cortex. Again, it's back here in red on that extreme posterior tip of the occipital lobe, and it receives visual information from the receptors in our retina. The visual association cortex, all here in this purplish color, this surrounds that primary visual cortex here in red, and it uses past experiences to interpret visual stimuli, so color, form, or movement. An example again is that ability to recognize faces. And complex processing is going to involve the entire posterior half of the cerebral hemispheres. Next, moving on to olfaction and gustation. So sensory info from our olfactory sense receptors. So we have our olfactory receptors in the olfactory bulb. They're going to go to the olfactory cortex. And the olfactory cortex is located on the base of the frontal lobe and the medial aspect of the temporal lobe in an area referred to as the uncus. So you can see, it doesn't really show you it in this one, but the uncus is going to house the olfactory cortex, which parts of it were once part of that rhomboencephalon, and includes part of the amygdala, which is part of the brain that's located with emotions and memory. And we're going to talk more about the limbic system later. The olfactory cortex, which is more what you're seeing, you can see the olfactory bulb right here, your olfactory tract, and then you see this gustatory cortex down here in the insula region. But that olfactory cortex is going to send information from the insular lobe, which again is deep, right? Which also receives taste input from this gustatory cortex. So you've probably heard before how we kind of mix smelling and tasting together to get flavor. That's the reason why. They're both located here in this insular portion of the brain. So our gustatory cortex, again, is in this insula. just deep to the temporal lobe. It's involved in the perception of taste. We also have a visceral sensory area. This is posterior, so kind of right behind this gustatory cortex, and it is involved with conscious perception of visceral sensations, such as an upset stomach or a full bladder. So again, your receptors for olfaction and taste are going to detect different chemical stimuli. They transmit to your insular region, right, for your taste, and the uncus for your smell, and that's linked with memory and emotion. So next we're going to look at touch and spatial discrimination. So our touch cortex is considered the primary somatosensory or touch cortex that resides in that post-central gyrus. There we can tell the difference between things like temperatures, pressure, vibration, etc. This also gives us proprioception, so in other words, knowing where our body is in space. So in other words, being able to close your eyes and touch your nose, right? That's your proprioception. Your primary somatosensory cortex is working when you do that. In terms of spatial discrimination, so knowing that you are feeling something in your hand and not with your foot, that is what spatial discrimination is. And our association area is going to help us understand things like the size and texture of whatever it is that we are touching. So when the association area is working, you can reach into your pocket, if you have a pocket full of coins, and you can tell which coin is a quarter, a nickel, or a dime. And damage to the somatosensory association area on that parietal lobe behind the primary somatosensory cortex is going to leave sensitivity to touch unaffected, but it disrupts the ability to recognize objects by touch. So you can see here, talked about this post-central gyrus here. So that's where we're going to be dealing with our touch and our spatial discrimination. So this is kind of considered your touch cortex right in here where this bright green is. And this is one that wasn't shown in those last pictures, looking here at your equilibrium and your balance. So you can see here this vestibular cortex that's in the insula region, right? So the vestibular cortex, it's the posterior part of the insula and the adjacent parietal cortex. And it's responsible for conscious awareness of balance, so your position of your head in space. So here, looking at our posterior association area, this is a large region that's going to be located in the temporal, parietal, and occipital lobes. And it plays a role in recognizing patterns and faces and localizing us in space. And it's involved in understanding written and spoken language. And you can see that Wernicke's area, outlined by these dashes, is going to be part of that. It is within that posterior association area. So this is going to receive input from multiple sensory areas. It sends outputs to multiple areas, and it allows us to give meaning to information received, store in memory, and then tie it to a previous experience and decide on how we want to react to that. Then lastly, we have our limbic association area. And the limbic system is going to be involved in things like our sensations, our sensations, thoughts, emotions. Pretty much it makes us who we are. So this limbic association area provides emotional impact and a sense of danger. And it's concerned with a lot of behavior and emotion, but also, as you can see, it's really close to the prefrontal association area, right? So you see this limbic association area, it's right down here. And we talked before about this prefrontal association area. And I talked about how in some people that are kind of like daredevils, they'll just jump out of a plane without even thinking they're going to die. So this is right up here in this prefrontal association area, but then it's really close to your limbic association area. And it kind of makes sense that they're right near each other because they're both associated in better judgment, and by sensing danger, it's going to transmit that information through this prefrontal association area to tell the person to inhibit themselves from doing a certain thing. So having these two areas adjacent to each other contributes to what we know about its function, right? Because that limbic system is going to provide that emotional impact, kind of makes a scene important to us, helps us to establish our memories, helps us to kind of decide. It's concerned with our behavior and our emotion, right? So the limbic system provides most of our emotional-based drive and it helps to set the other areas of our brain into action. So I've talked about the anterior association area, the posterior association area, and the limbic association area. How do they all work together? So shown in this picture, I showed this picture to you before, you have a mom talking to the baby, right? So what's going on here? There are actually several areas that are acting together. So that anterior association area, again, is involved with intellect, cognition, recall, and personality, contains your working memory needed for abstract ideas, judgment, reasoning, persistence, and planning, and development of this frontal association area is going to depend on feedback from a social environment, such as this baby, feedback it's getting from its mom. The posterior association area plays a role in recognizing patterns and faces, so baby recognizing mom's face, right? Localizing us in space, and it's involved in understanding written and spoken language due to Wernicke's area. Then we have that limbic association area that we just talked about. It's going to provide emotional impact, makes a scene important to us, and helps us establish memories. So what's going on in this picture? Well, baby sees mom's face, kind of feels her emotion, hears the tone of her voice, feels hand on its head, notices a smile. Baby is then going to develop a memory of comfort in mother's arms and in seeing mother's face and hearing her voice. Baby learns speech from watching and listening to mom. The baby develops bonds and attachments from its mom, from this multimodal type of integration of that sensory information. So taking in that sight, the sound, touch, and smell. There was actually an interesting article that talked about looking at MRIs of babies' skulls when they were touched, or not of their skulls, looking at MRIs of a baby's brain when they were touched by their mom, or even when they were just, their mom kissed their head. And the amount of just all those neurons firing, it was just really cool. If you run across that article, definitely read it. Because it's just talking about how just a little bit of touch like that can really help in terms of development for a baby, because they're at such a critical stage in their life to start forming all of these different portions, all these different association areas. So I mentioned before that we have two of those cerebral hemispheres, right? And hemispheres communicate almost instantaneously via fiber tracts and functional integration. But each hemisphere has abilities that differ from the other. And you probably heard this before, whether you're right-handed or left-handed, you've probably heard, oh, you have that type of personality, or you're good at those things because you're left-handed or because you're right-handed. So each hemisphere has abilities that differ from the other. So cerebral dominance refers to the hemisphere that is dominant for language. And 90% of humans actually have left-sided dominance. Usually this is going to result in right-handedness. In the other 10%, they're left-handed and they have right-sided dominance. So functional specialization of each hemisphere is actually more pronounced in men because females have more connections between the two hemispheres. And the left hemisphere generally is going to do things like control language, math, and logic, whereas the right hemisphere is more for visual spatial skills, intuition, emotion, artistic, and musical skills. And again, those hemispheres they're communicating almost instantaneously via fiber tracks and functional integration. Excuse me. So that doesn't mean that if you're right-handed, that you're not going to ever be an amazing artist. That's not the case at all. I know many right-handed people that are amazing artists, right? Because we have those hemispheres communicating almost instantaneously. But sometimes you can see these scenes kind of playing out where those of us that are right-handed, we're going to have left-sided dominance, right? So generally more analytical, right? Math, decision-making, more, just said to be more analytical people, right? Whereas people that are left-handed, they're going to have right-sided dominance in their brain. They're said to be more creative type people. I actually have a sister who is left-handed, and she is very creative, amazing artist. I couldn't draw a stick person if my life depended on it sometimes. So I am right-handed, more left-sided dominance, much more into the analytical way of thought. And again, this doesn't always hold true. So you may be sitting there and like, well, I'm this-handed and that's completely opposite of what I'm good at. Again, we have that instantaneous, almost instantaneous connection of those hemispheres via the fiber tracks. So it doesn't mean that you can't be really good at something even if you have the opposite dominance. But just kind of something you probably heard about before, just kind of explaining it to you here with cerebral dominance. And as a reminder, each hemisphere, again, is going to be concerned with the opposite side of the body. So cerebral white matter is going to be responsible for communication between cerebral areas and between the cortex and the lower central nervous system. And these areas are going to consist of myelinated fibers that are bundled into large tracts. So the myelin sheaths that cover those axons are going to be classified according to the direction they run, whether it's association, commissural, or projection fibers. I know those are new words for you. We're going to talk about them and actually see them in the next slide. So here are those different types of fibers. So again, the white fiber tracks of the cerebral hemispheres that I just kind of introduced to you in the previous slide. And what's really cool is these are kind of color-coded to help you. So you see your association fibers, they're going to be in these pink colors. So association fibers are going to be horizontally running fibers, and they're going to connect different parts of the same hemisphere. So you notice how these little pink fibers, they're just connecting different portions of the same hemisphere. They're not spanning the two hemispheres, right? They stay in one cerebral hemisphere. Then we have our commissural fibers. These are horizontal fibers that connect the gray matter of two hemispheres. And they're shown here in green, and you can see how they're going from this hemisphere to this hemisphere, right? And they're connecting that gray matter, right? Connect across there. Then we have our projection fibers, and projection fibers are vertical fibers that connect the hemispheres with lower brain or the spinal cord region. So those are in purple here, and you can see how they're going from up here, down here to the spinal cord region, right? So those are your projection fibers. We also have this area here that I highlighted in yellow for you. So our internal capsule, which is right here, this is the projection fibers on each side of the brain stem are going to form this kind of compact band, and it's going to pass between the thalamus and some of the basal nuclei. We also have this corona radiata right here. That's going to be the projection fibers that radiate through this cerebral white matter to the cortex, right? So just a few areas there that I wanted to point out with your internal capsule right here and this corona radiata. Otherwise, you have those three different types of these white fiber tracts. Association fibers, they're going to be in the same hemisphere. Commissural fibers, they're spanning, connecting the two hemispheres. Then your projection fibers are going from your cerebral cortex down to this lower area near your spinal cord. And here you can see these different types of fibers. Again, these white fiber tracts of the cerebral hemispheres. Again, those association fibers in pink, they're going to stay in the same hemisphere. Your commissural fibers, they're going to connect between two different hemispheres. And then those projection fibers, they're going to go down from each hemisphere to the brainstem. And you may have heard of this before. This is your corpus callosum. This is pretty much this huge bridge that happens between those two cerebral hemispheres, and it allows for the connection between them. So it's a broad band of nerve fibers that joins these two hemispheres of the brain. It is the largest commissure, and it's responsible for transmitting neural messages between the right and the left hemispheres. Because remember I told you, maybe even with that cerebral dominance, maybe you are right-handed and a really good artist. Well, we have this corpus callosum area that actually allows very quick transmission of those messages between the right and the left hemisphere. So cerebral dominance doesn't always play true, right? You can still be really good at one or the other, even if it's opposite of what you should be dominant at. Okay, so I wanted to end this lecture by talking about this case of conjoined twins. You may have heard of this in the news. It was, oh goodness, about 7 years ago. So this is Jaden and Anais McDonald. They were twins. They were 13 months old, and they shared a 5 by 7 centimeter section of their brain tissue. About 80% of these, they're called craniopagus twins. Generally, 80% of the time they're going to die of medical complications by the age of 2 if they're not separated. So that's why at 13 months of age they were separated, and they're considered to be a medical miracle. This type, the craniopagus twins, this type of conjoined twins only occurs in about 1 of every 2.5 million births. And 40% of twins that are craniopagus, when they're born, they're stillborn. So doctors use these really complex 3D models, like the one that you see here, to see the connection of the brain tissue and the vessels between the two boys. The surgery lasted more than 27 hours, and they were separated again at 13 months in age. That was back in October 2016. The surgeons actually almost stopped halfway through because they found the boys shared more vessels and brain tissue than they originally thought. So what do you think would be the negative part of being conjoined, being craniopagus twins? Think about it. They're not able to develop their back muscles, so they can't really be on their stomach, right? There's going to be a crowding of space in the brain, probably seizures. How would you put clothes on over your head? You really can't, right? You have to go from your feet and go up, right? Unable to ride in a car seat or be carried or cuddled separately. So those are just some of those negative reasons, let alone the fact of the matter that 80% of craniopagus twins are going to die before age 2 if they're not separated. So again, the surgery was successful. They were able to separate them. There have definitely been some struggles since then. I actually put this article here. It was from about 3 years after the surgery, talking about how one of the boys is definitely very different from the other son. So kind of interesting to see the two different personalities, different strengths. So they shared 1.5 to 2 inches of brain tissue. And that outer brain tissue is what? It's the cortex, right? The association areas and the gray matter. So the inner neurons providing communication on what we feel and see and how we move. So surgery pretty much set them back to infancy in terms of things like speech and their gross motor skills. And Anais, one of the boys, faced major setbacks over that first year. He stopped rolling, he stopped playing with toys, he stopped lifting his head because so much fluid had built up around his brain and started to weigh him down. He's unable to speak. I don't know if that's still the case. This is kind of the most recent article that I could find from 2019, but just kind of things to think about in terms of how important that cortex, our association areas are, because one of these boys is doing pretty well, the other is really struggling with a lot of things that we take for granted, just being able to do really easily. So today we learned first about the motor. Well, first really we learned about the development of our amazing brain from that neural tube to the adult brain. Then we talked about the motor parts of the brain and the sensory parts of the brain, as well as those very important association areas. So that is all for this lecture on 12A. I will see you next time for our lecture on 12B. Mahalo.